Now there is competitive inhibitor. What is that? Now when the inhibitor closely resembles the substrate in its molecular structure, so inhibitor and substrate, they closely resembles in molecular structure and inhibits the activity of the enzyme. They confuse the enzyme. Now it is known as the competitive inhibitor. Now due to its close structural similarity with the substrate, now inhibitor competes with the substrate. Now they are similar, okay. Now they can get to the enzyme, so for the substrate binding site of the enzyme, now consequently the substrate cannot bind, and as a result the enzyme action declines. Now inhibition of succinic dehydrogenase, it is an enzyme, okay. So it is an enzyme. Malonate, it is a substrate. No, it is an inhibitor, and substrate is the succinate. Now competitive inhibitors are often used in the control of bacterial pathogens. Now classification and nomenclature of enzymes. Now there are thousands of enzymes present, and enzymes are mostly proteinaceous, okay. Now these enzymes are divided into 6 classes, each class with like 4 to 30 subclasses, okay. Now they are numbered by a 4-digit number. Now first one is the oxidoreductase, that is dehydrogenase muscle reduction oxidation occurs, okay. So oxidoreductase and dehydrogenases. Then second class is transferases, like transferring of a group like other than the hydrogen, okay, between the substrate and S dash. Now hydrolases, it is a third class. Now these enzymes hydrolysis of ester, ether, peptide, glycosidic C bonds, C halide or PN bonds, okay. Now fourth is lysases, enzymes that catalyze removal of groups from substrate by mechanism other than hydrolysis, leaving double bonds. So third is the hydrolysis, but lysis is other than hydrolysis that can remove groups and leaving the double bonds. So these are lysases. You see in the diagram CC XY, then gets XY is connected and C double bond is formed, leaving the double bonds. Now fifth one is isomerases, right? Isomerases, now including all enzymes catalyzing interconversion of optical, geometric or positional isomers. These are isomerases. Now seventh one is no sixth one is ligases. Now ligases, these are ligase, it means ligate. It can connect, it can join like two different compounds, okay. CO CS CN and PO bonds, CO CS CN PO bonds, right? Now cofactors. Now what are the cofactors? Now these cofactors very important, right? mm, no cofactors, okay. Okay, you see in the summary, the closer examination reveals that the relative abundance of carbon, hydrogen, and oxygen is higher in living systems when compared to inanimate matter, that is non-living. Okay, amino acids, monosaccharide, disaccharide sugars, fatty acids, glycerol, and fatty acids, glycerol, and nucleosides, nitrogen bases are some of the organic compounds seen in living organisms. Nucleic acids carry hereditary information and are passed on from parental generation to the progeny. Enzymes lower activation energy of reactions and enhance greatly the rate of reactions, right?